Welcome to part 3 of chapter 9, where we are going to talk about G protein coupled receptors. G proteins are molecular switches, and they are oftentimes used to regulate other pathways, either to turn them off or what we call down regulate. Decrease their signaling power. They are sometimes also used to amplify signals as well. They use a two-step process for signal amplification, and they're oftentimes used in hormone responses, and they are used also in in responses in which the cells need a very rapid reaction, especially when there's a low signaling concentration. So let's take a little bit closer of a look at G protein coupled receptors, or also known as GPCRs. They're very common in eukaryotic cells. They're not found in prokaryotic cells. They are found in plants, though, and they are found primarily in mostly hormone responses. The protein itself, known as a G protein, the receptor is the G protein, and it contains 7 transmembrane segments, meaning there are 7 polypeptide chains that make up this protein. It is a really big protein. The receptor is located in the plasma membrane. The external side is where the signal will bind to the receptor, and the intracellular side, a whole bunch of activity is going to occur in order to activate the G protein. G protein releases GDP and will bind GTP. This is similar to what we saw in the RTK pathway in that RAS protein. That RAS has a GDP attached to it, and when it gets phosphorylated, releases GTP. GTP will bind and cause a conformational change to the G protein. When this occurs, the G protein splits up into a couple of pieces. It's called dissociation, and it will dissociate into 2 subunits, an alpha subunit and a beta gamma dimer. The alpha subunit and the beta gamma dimer are going to interact with other proteins and cause a few different things to happen. And that's where we actually start to see the signaling pathway. Up until this point, we have simply just looked at activation. The signaling molecule, however, that bound to the receptor to begin with, eventually will break down and degrade. Most of the hormone signaling ligands that GPCRs use are very unstable, so they don't stick around for a very long time. And when this occurs, when the signaling molecule is no longer bound to the GPCR, then the alpha subunit will break off that phosphate group and off of GTP, and it will become GDP. When that happens, the whole process gets reversed, and those 2 subunits will come back together and bind to the G protein receptor, and the pathway is reversed, is turned off. So this is a little easier understood if we take a look at some diagrams. So first, let's look at the activation of it. Here is our GPCR, right here, and here is our signaling molecule in green. The signaling molecule will bind to our G protein coupled receptor. When it does this, this receptor is going to change shape. That's going to cause this G protein here, right? So you see this G protein is embedded in the membrane right next to the receptor. When this receptor tail on the intracellular side pops down due to conformational change from this ligand binding, these proteins are going to move over. They're going to move over, and they are going to attach to our GPCR. This causes a conformational change in this protein, and you can see this little GDP molecule with only 2 phosphate groups attached to it. Well, this free phosphate group is going to come in here, and it's going to bind to GDP. This is going to get released because of this binding action here. This gets released, and then GTP is now free to bind. When GTP binds, our beta gamma protein over here releases. Here is our alpha subunit, and our G protein receptor is now activated. So there's quite a few things that have to occur. We have the signal binds to receptor, receptor changes shape, activates G protein. G protein has GDP, separates, and GTP binds, and we now have our 2 subunits here. This GDP to GTP, this is why this entire process is called a G protein coupled receptor and why this is known as a G protein. My videos are not working, so I will upload the videos separately into D2L so you guys can view these on your own. Now, ligand-gated channels. In ligand-gated channels, these are found in both plant and animal cells. They sometimes are found in a few prokaryotic, but mostly they're in eukaryotic cells. These are pretty straightforward receptors. So they're usually channels of some kind. Animal cells use them for signaling between nerve and muscle cells. They're used between nerve cells. They're also used in heart cells for calcium ion exchange. This changes the membrane potential, thereby causing your heart to beat, so we can coordinate the heartbeat. So these ligand-gated channels, they play lots of roles in membrane potential electrical signals and charges across a membrane. So here's our signaling molecule, and the signaling molecule will bind to a region of this protein, usually more than one. Oftentimes 4, even 5 or 6 are required. As these signaling molecules bind to these proteins, it causes a conformational change in the shape of this protein to open up and allow the free exchange of some other molecule across the membrane. So we just need a bunch of these molecules here to bind, the signaling molecules to bind, and when the signal molecules bind in a high enough concentration, the channel opens up and other molecules are able to flow freely across the membrane. So think about your heart beating. Your heart beating is an electrical signal. So ions are exchanged across the heart muscle in what's known as the sarcoplasmic reticulum, which is a specialized part of the endoplasmic reticulum. And in that sarcoplasmic reticulum, we have ions of one charge will build up outside of this, this ligand-gated ion channel. And when that concentration of ions gets high enough, a signaling molecule will bind to these channels here and open them up so that all of these ions flood into the sarcoplasmic reticulum, causing a rapid change in electrical signal, which causes the muscle cells to contract. So that's how our hearts beat, is because of these ligand-gated ion channels. That's how fast they can work. Now, one other thing we need to look at are some intracellular receptors. All 3 of the receptors we have looked at right now are involved in extracellular signaling. So ligands coming from outside of the cell binding to these plasma membrane bound proteins or receptors and making the cell do something. But we also have receptors that are found in an intracellular platform. When they're intracellular, it's usually because a signaling molecule has the ability to freely diffuse across the plasma membrane. And the receptor for it is found in the cytosol as opposed to found embedded in the membrane. Now, one of these types of signalings is estrogen. Estrogen is a hormone, and it does actually cross the plasma membrane of the cell. And so the estrogen and its receptor form a complex in the cytosol. They become a dimer. In this case, it would be a heterodimer. This dimer estrogen complex will eventually move into the nucleus, and it will bind to DNA in order to activate the transcription of other specific genes. And transcription factors will regulate which genes are turned on and which are turned off. So the estrogen receptor becomes a transcription factor oftentimes when it binds in the cell. This is a form of gene regulation that is referred to as direct gene regulation. So here we have an example. Here is our estrogen receptor. Right? And here's over here on the left is what an estrogen molecule looks like. So estrogen flows freely, right? It kind of just diffuses across the plasma membrane pretty easily, and it's able to, because it can move across this plasma membrane, it enters into the nucleus. So this estrogen receptor is found inside of the nucleus. When it binds to estrogen, when estrogen comes into the nucleus and binds the receptor, multiple estrogen receptors get bound. They dimerize together, and in doing so, they turn on or attach directly to DNA. They cannot attach to this DNA until they're bound to estrogen, and two of them are bound to estrogen at the same time and then bound to each other. They then bind directly to the DNA and initiate transcription. That transcription will ultimately cause the synthesis of a protein that's going to do something to the cell, make it divide, make it produce something else, make it do something, right? So it's going to change the cell. Now with auxin, we have a different type of regulation. Now auxins are plant growth hormones, and auxin is going to bind its receptor here in the cytosol. So here's our auxin, these little blue molecules. They pass freely across the plasma membrane, and they are binding to their receptor in the cytosol. This receptor is then allowed to go through a nuclear pore, right? So this receptor bound to its signaling molecule is now of the correct shape to be allowed into the nucleus through a nuclear pore. This guy moves in through a nuclear pore, and when he moves in through a nuclear pore, he binds to another protein that's already sitting on the gene waiting to be told what to do. And so when these two come together and our ligand-bound auxin receptor binds to this transcription factor here, we start seeing transcription. The mRNA molecule for auxin moves out and becomes a protein that's going to initiate, remember, it's going to initiate the elongation of those cells. So the cell, it causes the cells to change shape and eventually divide. So it's going to increase cell density on one side of the plant as well as the length of the cells, causing the plant to bend. Remember that from the part 1 lecture, the example of auxin. So in signal transduction and response, sometimes we have what's called a two-component system. And in a two-component regulatory system, we see this in bacteria, fungus, and plants, but it does not occur in animal cells, but only in prokaryotes, fungi, and plants. The first component is called a sensor kinase. It's some type of enzyme-linked receptor that'll recognize a signal. And instead of, when it hydrolyzes ATP, recruiting a bunch of relay proteins, it actually will break off ATP and phosphorylate itself. This phosphate group will be transferred to what's known as a second component response regulator. The response regulator, the second part of this whole process, will directly regulate the expression of multiple genes, especially in prokaryotes. So instead of this big long kinase type cascade, we just simply have two proteins involved in this signaling pathway. So that's why it's called a two-component regulatory system. Now, what we really start to see in GPCR pathways is something called second messengers. All of our pathways that we have looked at have the ability to cause some kind of cellular response, but sometimes we need that cellular response to be amplified. So in second messengers, our initial signaling molecule binds to the surface of a cell and that receptor that's on the surface of the cell, and it's referred to as the first messenger. And if just that signaling molecule is all it takes, like in the EGFR pathway, to make things happen, then we're looking at a one-component system. But oftentimes we don't have a high concentration of initial signaling ligand, so we're going to have to amplify the signal to really get the response that we want. I might want to talk to a crowd of 100,000 people. I'm pretty loud, but I don't think 100,000 people can hear me. So I'm going to use a megaphone or a microphone to amplify the sound so that everybody can hear me. And that's really what's going on with second messengers. The cell is, the initial signaling is not enough, and the cell needs to amplify that signal to really get the point across to the rest of the cell that something has to happen. So a lot of different transduction pathways use and lead to production of second messengers. And some examples of these are cyclic AMP, calcium ions, and diacylglycerol, and what's known as inositol triphosphate, all involved in some heartbeats. So let's take a look of what's happening in signal transduction with a second messenger. Cyclic AMP, known as cyclic adenosine monophosphate, or CAMP for short, is known as a second messenger. And what we have here is a GPCR pathway, right? So here's our signaling molecule, here's our G protein coupled receptor. Here's the G protein that it's coupled to. When this initial molecule, right, our primary molecule, binds to this G protein receptor here, it activates the G protein itself. Now, when the G protein gets activated, here is the beta gamma dimer, but here's the alpha. And the alpha domain moves over and passes the beta gamma. It moves over across the cell membrane to an enzyme or protein known as adenylyl cyclase. Adenylyl cyclase is going to take ATP molecules and start building what's known as CAMP, or cyclic AMP. Cyclic AMP is going to then become a second messenger. And cyclic AMP takes part in intracellular signaling, and it starts activating what's known as PKA, or protein kinase A. Protein kinase A gets activated when cyclic AMP binds to it. And when it binds to it, it starts activating other PKAs. And these PKAs start phosphorylating other proteins, and they convert ATP into ADP. I know this sounds very confusing and it's really intense, but just remember, cyclic AMP is working as a second messenger, so we're restarting a new transduction pathway. So my first pathway is my G protein. My G protein activated adenylyl cyclase. Adenylyl cyclase activates CAMP, produces CAMP to activate protein kinase A, which basically just runs around the cell and turns on light switches and starts activating all kinds of other proteins. But once CAMP has turned everything on, it's amplifying the signal because that adenylyl cyclase just keeps pumping out. As long as that alpha subunit is attached to adenylyl cyclase will continue to produce cyclic AMP. And the cyclic AMP just keeps on coming, and it keeps on activating more and more of those protein kinase A's. So when we need to turn it off, we have to turn off the G protein coupled receptor. We have to go all the way back to the very beginning and take that ligand, that primary ligand or primary signaling molecule off the G protein coupled receptor. This is then going to cause that alpha subunit to dissociate from adenylyl cyclase and match back up with that beta gamma dimer. If adenylyl cyclase is turned off, then we will no longer be producing cyclic AMP. And an enzyme known as phosphodiesterase is going to start breaking down cyclic AMP or CAMP into AMP. If no CAMP is being produced, then the levels of it will fall, which means the activation pathways for protein kinase A will also decrease. So PKA starts to become disactivated or inactivated. If the PKA is inactivated, then the proteins or enzymes they were responsible for turning on will now start to be turned off. So it's the exact same process as activation. We're just turning it off. We're just moving in the opposite direction. So here's turning off of CAMP, right? So these phosphate groups coming back here, and we no longer have our CAMP levels get really low, so phosphodiesterase starts breaking down CAMP. We also have this signal molecule coming off, so our alpha subunit has come back over and joined with our beta gamma, and that means that adenylyl cyclase is no longer producing cyclic AMP. Now, why do cells use this? Why do they use this second component here? Why are cells using this second messenger? And one of the primary reasons is signal amplification. CAMP in particular has two very distinct advantages. One is it can amplify a signal intensely. If you take a look here, I have one G protein coupled receptor bound. This G protein coupled receptor, when it activates adenylyl cyclase keeps pumping out CAMP, right? So this guy was only activating one, but in activating one, this guy here starts producing lots and lots of these CAMP molecules, and every CAMP molecule activates a protein kinase A. And protein kinase A, each one of these can turn on and start phosphorylating. Remember, it's a kinase, can start phosphorylating multiple transcription proteins. So these purple proteins are all targeted. They need to be turned on, and we need tons of them. And if we need lots and lots of these purple proteins activated, the quickest and easiest way to do that is through a second messenger. Now, the second advantage here is speed. How fast can we get a cell to do something if we amplify the signal intensely? So there was an experiment done where enough CAMP, right? They were able to measure enough CAMP after 20 seconds to be able to read a signal. So here we have a nerve cell in brain tissue, and over here they added, I think it was some serotonin. Yeah, they added serotonin to the mix, and as soon as serotonin, within 20 seconds, cyclic AMP was being signaled throughout the cell. Look at all of that cyclic AMP. This is all the result of cell signaling, this change in color. So 20 seconds, they can start changing what a cell is doing and producing. All right, we're going to stop here, and we will move into signal transduction by calcium ions because this starts getting into cardiac muscle contraction. So we will move into this in part 4 of cell communication.